the Hendersen-Hasselbalch equation. One thing that becomes clear straight away is that if the term in brackets equals 1, the log becomes 0. In other words, if there is an equal concentration of acid and conjugate base, pH equals the pK of the acid. It is interesting to see what happens to the pH of a solution of a weak acid if we gradually add small volumes of strong base until the full molar equivalent of the weak acid has been added. In the lab, this is called a titration. Using the same maths as before, we can calculate that a 100 millimolar solution of acetic acid will have a pH of 2.88, meaning that an even smaller proportion of acid is dissociated, only just over 1%. If we add a little bit of strong base to shift the proportions of acetate to acetic acid, the pH jumps up. Adding still a little more, the pH increases again, but by a smaller amount. Now, if we progressively shift more acetic acid towards deprotonation, we soon reach a point where both concentrations are the same. This is the point where, as we have said before, the pH equals the pK of the acid. Note that this is the flattest bit of the curve. If we then continue to add base, at some point the pH rises more steeply again. Quite a few interesting things can be learned from this curve. First, the titration curve is flattest when the pH is near the pK. Around that time, the acid has the strongest buffer capacity, meaning it is able to resist changes in pH when acids or bases are added. Right at that midpoint of the titration, the concentrations of acid and conjugate base are the same. The issue of buffer capacity is one that is very important in the biochemical lab. Bottles with buffer chemicals will sometimes say on the label what the pK is. And that means it can be used if you want to buffer, that is, stabilize the pH of a solution in your experiment at a pH no more than 1 pH unit above or below the pK. Here, for example, you can use this buffer for pH values between 6.5 and 8.5. So, to wrap up, in a titration curve of a weak acid, the protonated form is dominant at low pH. Near the pK of the acid, roughly equal concentrations of the acid and its conjugate base are in solution. This is also called the midpoint of the titration. Within about 1 pH unit either side of this pK, the acid will buffer effectively. At a pH well above the pK, a weak acid is mostly deprotonated. The concentration of the conjugate base is bigger than that of the undissociated acid. Now that we understand those titration curves, we can have a brief look at some related cases. First, a weak base can be titrated with a strong acid, and we get sort of a mirror image of the weak acid titration curve. In this case, the midpoint represents the pK value of the conjugate acid of this weak base, which will be above 7. Quite a few acids with biological relevance can dissociate more than one proton, including phosphoric acid here. The titration curve for such polyprotic acids contains 2 or more of these midpoints, and each of them represents 50% dissociation of the first, or second, or third proton respectively. You can see that phosphoric acid has 3 pK values ranging between 2 and 12. Finally, some biomolecules, in particular the amino acids, have functional groups that mean they can act as both proton donors and acceptors. Glycine, for example, has both an acidic carboxyl group and a basic amino group. A titration curve of glycine shows 2 pK values, one in the acidic range, which is due to the carboxyl group, and one around pH 9, near the pK of the amino group. More about that in the next lecture on amino acids. Molecules that can both donate and accept a proton are sometimes called amphiprotic or amphoteric.